There are different processes that take place in our bodies without even noticing them, such as embryo development, where a zygote developing from the union of a sperm and the ovum develops completely into a fetus or a human being. Or when we grow up from little kids to adult people, or when our cells repair themselves after breaking a bone or cutting ourselves. All these processes and others are due to a single process that is called mitosis. Now, what is mitosis? Mitosis is the division of a diploid cell, 2N, to produce two genetically identical daughter diploid cells, 2N. We're going to discuss later the word diploid and the term or the value 2N to understand what we mean by them. But the main point here is that mitosis is a cell division, it's a type of cell division where the cells divide or multiply to give genetically identical daughter cells. So the cells resulting from mitosis are identical, they are the same. The importance of mitosis, as we said earlier, is, for example, it permits the growth and development of a multicellular organism, such as humans, for example. It grows, or we humans grow by mitosis. Our cells divide using mitosis to allow our body to grow up. The other importance of mitosis is that it allows the replacement of damaged or dead cells. If we want to understand mitosis, we have to get to know its steps or phases. First of all, we have to be familiar with a term called cell cycle. All cells in our body pass through a life cycle, or what we call a cell cycle. This cell cycle is divided into two main parts, interphase and mitosis. Now, interphase is a separate phase from the cell cycle. We might talk about it later in another video. And now what we're concerned of is mitosis. Mitosis is divided into four phases. The first phase is called prophase, the second one is metaphase, then anaphase and telophase. Of course, we're going to get to know each phase alone later on in this video. And if we want to represent the cell cycle, we can represent it like this. And as we know, the word cycle means that it repeats itself. So it starts, if we want to say by interphase, most of the cell life is interphase, and there is the cell division or what we call mitosis. Now let's start with interphase. Of course, we're not going to talk in details about interphase, but in order to understand mitosis, we have also to understand interphase. During interphase, if we look at a cell under the microscope, we can see, of course, this is a diagram, we can see the following main features. First of all, we can see the nuclear membrane. The nuclear membrane is clearly visible during interphase. Moreover, there is in the nucleus what we call chromatin and not chromosomes. So the genetic material, the genetic information or material is in the form of chromatin, which is a tangled filament, if you want to say, or fibers that is not condensed. We can't see chromosomes as its form. Okay, we're going to talk later on about chromosomes. Moreover, this phase, which is called interphase, is the phase of cell growth and DNA replication. And we are going to talk about this later on. We will understand why should a cell pass through interphase and make what we call DNA replication. So if we want to look at another representation, we can see the cell like this. It has the cell membrane intact, okay, and inside it there is the chromatin that is a tangled fiber and it's not condensed. We can't see chromosomes, etc. So this is the first thing, the first phase in the cell cycle, which is interphase. And bear in mind here, we didn't start mitosis yet. Now we'll start with mitosis. The first phase of mitosis, as we said earlier, is prophase. Now let's look at the same cell under the microscope. So this is a cell during prophase. What happens during prophase, first of all, is that the chromatin that we saw earlier, which is the tangled fiber, condenses into chromosomes of two chromatids. Okay, so what is this condensation? What is this? We have the chromatin here, okay, which is the genetic information or material. It starts to condense through a complex process. It starts to turn around itself and condense and form what we call a chromosome. It makes this shape, which is a chromosome, and this chromosome is made up of two chromatids. Each one of these is called a chromatid, and they are attached at a certain position called the centromere. Okay, so this is the chromosome. It was during the interphase, it was a tangled fiber, which is called chromatin, but during prophase, it condenses to form chromosomes, and it has to form this shape because this shape allows the cell to divide. If the genetic information or material remains as chromatin, the cell cannot divide. The next feature about prophase is that we start to see what we call asters, the centrosomes or centrioles, which are dormant during interphase. They are not functional. During prophase, they start to change into what we call an aster, which is like a star-shaped structure. Moreover, the nuclear membrane disintegrates or starts to disappear, to break down. So the chromosomes now are free in the cytoplasm of the cell. They are not found in a closed nucleus anymore. They are free. And finally, we start to see some fibers, some filaments. They are made of protein, and we call these spindle of fibers or the achromatic spindle, which starts to form during prophase. And we will see the importance of these spindle fibers later on in the next phase. So during prophase, first of all, the nuclear membrane disappears. We see the chromatin condenses into chromosomes, and the centrosomes become asters, and we see the spindle of fiber that form between these asters. The second phase after prophase is metaphase. Now this phase is very special, it's very important. So what happens during metaphase? The chromosomes, first of all, that we saw earlier forming in prophase, they move to the center of the cell and line up at or along the equator. If we look at the cell, it looks like the planet Earth. There are two poles, and in the middle there is the equator here. So the chromosomes come and line up at the equator, okay? And here, when they line up at the equator, this is called the equatorial plate. They form what we call the equatorial plate. Moreover, the centromeres, which we said earlier that they attach, the two sister chromatids are attached at the centromere, the centromeres attach to the spindle of fibers, okay? So the spindle of fibers, of course, the asters become at opposite poles of the cell, okay? One at one pole and the other at the opposite pole, and the spindle of fiber is between them. And the chromosomes, which align at the equatorial plate, will attach to the spindle of fiber. Each one will attach to a fiber using their centromere, okay? So another explanation here, we have the chromosomes. They will, of course, first the asters will move to opposite pole. Here we see the spindle of fiber, and the chromosomes now will line up at the equator and attach using the centromere. They attach to the spindle of fiber like this, okay? So they are at the equator, at the middle of the cell. This is metaphase. The main feature of the metaphase is the formation of the equatorial plate. Now the third phase is anaphase, and this is also a very important phase, and it has all the action in it. What happens during anaphase? Earlier we talked that the chromosomes line up at the equator. During anaphase, something very special happens. The centromeres break down, and now the chromatids of the same chromosome are separated. Now what do these chromatids do? The centromeres divide, and sister chromatids of each chromosome separate. And they don't only separate, they move to the opposite poles. So let's see here, for example, this chromosome. The two sister chromatids of this chromosome separate from each other, and each sister chromatid will move to the opposite pole. The same thing for the other chromosomes also. Okay, see how every sister chromatid will move to an opposite pole. Okay, we call this polar ascension. This phenomenon or this step is called polar ascension, which means movement to the poles. Okay, here what happens after polar ascension? We will get two sets or two groups of chromosomes of one chromatid. Okay, we will have two sets, two groups. After they were only one group together, now we have two sets of chromatids, and these sets are identical. Why are they identical? It's easy. Let's see this chromosome which had two copies or two sister chromatids which are identical. Now each sister chromatid moved to one pole and so on. So this set is identical to this set, as we can see. Let's explain it furthermore. Okay, here we have the chromosomes that are lined up at the equator. What happens is that each sister chromatid of each chromosome moves at the equator. So if we want to simplify the idea, the red chromosome, the big red chromosome, we have one sister chromatid of the big red chromosome in one pole and the other sister chromatid. Okay, now let's see now the small blue chromosome. This is one chromatid here and one chromatid at the opposite pole. The small red chromosome, here one sister chromatid to the pole and another sister chromatid to the other pole. Okay, and finally the big blue chromosome, here one sister chromosome to one side or one pole and the other to the other pole. If we compare the two sets of chromosomes we have, we will see that they are identical to each other. Now if we take a look to the cell during prophase and the cell during anaphase, at the end of anaphase, let's look at the chromosomes, their aspect. We see that during prophase we had 4 chromosomes in the cell, but each chromosome is made up of 2 chromatids, while at the end of anaphase, in each set we also have 4 chromosomes, but this time each chromosome is of 1 chromatid. Here what we talk about the ploidy of the cell was 2n equal 4. It had 4 chromosomes diploid. Why does it have 4 chromosomes, or why is it 2n? Because in this cell, for each chromosome, there are 2 homologous copies, or for each chromosome, as in any living thing, there is, for example, one from the mother and the other from the father, and here one from the mother and the other from the father. That's why we have, or a cell has, 2n of chromosomes. Okay, 2n means 2 sets. So it was diploid because it's 2n, and during or after anaphase it's still 2n, because look here, there is still a copy of the chromosome, for example, from the mother and from the father, and another chromosome from the mother and another chromosome from the father. Okay, there are still 4, but the difference here that the chromosome during prophase is made up of 2 chromatids, while after anaphase it's now made of 1 chromatid. The last phase during mitosis is telophase. It's the phase that wraps up everything, that finishes the mission. During telophase, after we got the 2 sets of chromosomes separated from each other, now we have to separate the cell. The cell itself has to break into 2 cells. So what's the main feature during telophase is the breaking down of the cytoplasm of the cell. But first, things have to return to what they were before mitosis. So the spindle fiber breaks down, we don't see any spindle fiber, and the asters return to being centrosomes, they return to their previous state. Moreover, the chromosomes decondense again to become chromatin because they are done dividing. Division is done. They have to return to their previous state, which is chromatin, and a new nuclear membrane forms. We notice that there is a new nuclear membrane to enclose the chromatin. They shouldn't stay in the cytoplasm. Okay? And finally, and most important feature of the telophase is what we call cytokinesis, where the cytoplasm is cleaved and gives us two entire or complete cells, daughter cells. Of course, there are new organelles that form. Each cell will have its organelles, the mitochondrion, the endoplasmic reticulum, the ribosomes, etc., etc. And these cells, of course, are identical. Why are they identical? Because the sets of chromosomes each have are identical. So this is telophase. Let's check it again. We have two sets of chromosomes identical at each side. Okay, the nuclear membrane forms again, and we have cytokinesis here in the middle in order to get two separate cells. Okay. And of course, the chromosomes will decondense into chromatin. The chromosomes of the daughter cells at the end of the mitosis are simple, meaning they have one chromatid. Okay, as we saw here, notice that the chromosomes are made only of one chromatid. Now, this is a problem. Why? Because could the daughter cell perform another round of mitosis? Let's look at this daughter cell and this daughter cell. If we are talking, for example, about the development of an embryo, the formation of a fetus, the cell undergoes millions of mitotic divisions. The single zygote starts to divide continuously over and over again. But let's imagine this cell, it wants to divide to other two cells. Can it do mitotic division in this state? Of course not. Why? Because the chromosome is made of one sister chromatid. When it wants to divide, there is no other sister chromatid that separates. Here comes the importance of interphase. The solution is by interphase. Why? Because during interphase, after doing mitosis and finishing mitosis, okay, the cell here is made only of one chromatid. It undergoes what we call DNA replication. Replication means duplication. Okay, so each chromatid will form another copy, another identical copy of it. Okay, so the chromosome becomes again made of two chromatids, and this allows the cell to make a new mitosis, because the cell cannot divide if the chromosome is made of one chromatid. It has to be made of two chromatids, two identical sister chromatids. So due to DNA replication, the chromosomes will become made of two chromatids again, and they will be a part of a new mitosis to give two daughter cells. Again, each daughter cell will pass through another interphase to make another DNA replication, duplication, to allow it to make a new mitosis, and so on. Now, someone might ask, is there a difference between mitosis of animal cells and plant cells, or do they undergo mitosis in the same way exactly? Of course, plant cells, since they grow and develop and reproduce, of course, they also undergo mitosis. But between plant cells and animal cells, there are slight differences. The first difference between animal and plant cell is that animal cells have asters, which are previously centrioles, okay? The centrosome or centriole, they have them, but the plant cell don't have asters. So when plant cell undergo mitosis during anaphase, there is no aster that will carry the spindle of fiber and guide the chromosomes during their division. The other difference is that in the animal cell during telophase, there is the formation of the furrow, which cleaves the cytoplasm of the cell to get two daughter cells, while in the plant cell, due to the presence of the rigid cell wall, there is no furrow, okay? No furrow, no cleavage take place, but the formation of a new cell wall between the two cells forms. There is a formation of the cell builds a new cell wall between the two daughter cells. Here we come to the end of our video. Thanks for watching. Don't forget to answer the quiz in the description to test your knowledge about mitosis. See you later in a new video. Goodbye.